Hello everyone. So I'm going to make a video. This is about the unknowns, that assignment you're going to be doing in lab. So I'm going to click on the week 11 tab and unknowns this week. So if you're watching this video, you've already done this because it would have been at the top. So we have some documents, and then here is where I want you to turn in your unknown. Okay, when you've completed it, and there is a due date for it, but when you complete it, I want you to submit it right there on that link. So we have a little introduction. What we call this an unknown is where you're given a tube of bacteria. You don't know what it is, and we're going to run a few tests and try to approximate what it is inside the tube. And then you're going to write a short scientific paper on it. Scientific meaning you're basically going to answer questions in a certain order. It's not going to be a literature paper or anything like that. So instructions for completing the unknown. So I'm going to click on this, and then I'll show you the flow chart. And I will, of course, go over this in lab as well. So here it is. Every unknown is going to have a number. Assume it's a pure culture. That means it only has one organism growing on it. And you are going to follow aseptic technique. So the first thing you're going to do is a gram stain. And you want to, once you do the gram stain, of course, you want to look at it under the microscope. Determine if it's gram positive or gram negative. And then the next thing is you're going to write down the cell shape arrangement. You really can't get that wrong unless you put like triangles or rectangles, or you're going to have either rods or causi, and then you put what you think the arrangement is, and you go with the majority. It says, according to your gram stain results, determine which portion of the flow chart you'll need to follow. Okay, and then, in fact, maybe I'm hoping I can bring up an example of that in a second. It says you'll need to include the following information in your unknown report. So a heading, and I have an example for this, your gram stain results, your cell shape arrangement, the list of tests you performed, for each test, an explanation of the test. In other words, what's it positive and negative for, or what does that mean? If something is urease positive, or the urea turns pink, it means it's positive for the enzyme urease, and it broke urea down into ammonia, and then you got the color change. And then this part, I'm going to let you use the internet or Bergey's manual, anything you want to use in the library. You have to answer these questions about your unknown. Now, I wanted to, and I ended up making this too wordy. It's a previous example here, but I put it in blue. But here is says examples on how to cite sources. So anything that you use, please put the source of that. I gave you an example of a book website. That's another website there. But here's the format. So here is the title up here. It says 15% of your grade. There's the number, bolded, underline the number. Gram stain results, and anything in blue here is just comments I gave you to help you write the whole thing out. So list in order according to the flow chart of the tests that were performed. So you list them here, and then here's a description of the results. Okay, so there's EMB, the things you might add to it, the identification as to what you think it is. Once again, you go with the majority, and then the research questions. And I put in blue there, anything I hope that will help you. Now let me take a station break, and I'll be right back. Okay, we are back. And I had just wrapped up going through a really quick and short way of what you're going to be doing. You're basically going to be doing some tests, and I kind of went through and showed you where you can find the information on how to write up your paper. So that's one thing. Oops. And then here is a total sample unknown without the blue writing in it. So gram stain results, cell shape arrangement, list of tests performed, description of tests, identification of the unknown, research questions. Okay, and that's a good example of how to do it. Now, on this page, all that's under here. Now, the unknown flow chart. You're going to need this. So I'm going to do that. And then here we go. Oops. Here's the gram-positive side. Here's the gram-negative side. So we'll start here first. So when you're given your unknown in class, it's going to be a tube of bacteria, first thing you're going to do is a gram stain. So you can tell if it's gram-positive, gram-negative. And you want to write down the cell shape arrangement. Okay, the next thing you have to determine, okay, if your here's the gram-positive side, here's the gram-negative side. If you're gram-negative, you're going to run the same day. You're going to inoculate an EMB plate. You're going to stab one of those SIM tubes, little gel plugs. You're going to inoculate a broth tube. And you're going to inoculate a citrate slant. You're going to do that all at once. You're going to put it in the incubator. You're going to let it grow overnight or in 2 days. And then you're going to come back and you're going to see what changed colors and what did not. And remember, with the SIM, you have to add the stuff called Kovacs reagent. And it tells you if it's positive for indole, if a little red ring forms at the top. So here's an example. I'm going to go over another one as well. So here it is. Pretend this was your tube. Here's your gram stain reaction. It's gram-positive and it looks like little rods. Looks like they're in singles, but that's not important. The important thing is you gram stained your organism and it's gram-negative. And like I said, so you would then go to your flow chart, wherever that thing went. Here, you're on the gram-negative side. So like I said, you're going to streak a plate. You're going to stab one of these little SIM gel plugs. You're going to inoculate a broth, and you're going to streak an auger slant. Put it in the incubator and see what happens. So let's say you come back, and here are your results. Your EMB plate looks like this. It's got a green metallic shine. The indole, the SIM plug, once you add Kovacs, has a red ring at the top. Here's what the citrate looked like, and here's what the urea broth looked like. Remember, on EMB, dark purple colonies or the green metallic sheen, dark purple colonies means a positive for lactose fermentation. So does the green sheen. It means it's a coliform, but it's also a lactose fermenter. So on your chart, see, that's the first thing you would have noticed. If it was negative, you might start looking on this side. Well, you would start going on this side. If it's positive, you're going to look on this side. Clearly we have a positive, so the next thing we're going to look at is the SIM. Is it indole positive or negative? The SIM plug has a red ring at the top. That's a positive. Okay, same thing. You want to note that on this chart as well. You can either circle it, whatever you want to do with it. The urease, remember, was the salmon colored, which is this one. See, it starts out that color. That's a negative. If it was positive, it'd be fuchsia, hot pink colored. So urease is negative, so we're still going down this way, and it looks like the citrate as well. Remember, it's green. It is also negative. So our organism, after we look at all the tests, would be Escherichia coli. See, if it was positive and the indole was negative, the urease was negative, and the citrate turned blue, we would have had Klebsiella. Now, what happens if you have 3 tests that say it's here and we have 1 test over here? You go with the majority. Okay, so anyway, like I said, if this is positive, that's negative, but the citrate turned blue, that's 3-to-1. You still go with E. coli. Okay, let's do another example. Gram positives, okay? Let's say you do a gram stain, and this is what it looks like. You can see the blue, you can see the rods. It's a gram positive bacillus, rod shaped. Now up here it tells you if you have rods, you're going to be on this side. If you have cocci, you're going to be on this side. So the first thing you would do is a catalase test. You take some hydrogen peroxide, put a drop, see if it bubbles. And here we go. We got a positive there. Okay, it's positive for catalase. It means it produces catalase. It can split hydrogen peroxide into oxygen and water. Next thing you would look at is mannitol fermentation, which is a red broth tube. Remember, weak is orange, strong is yellow. So you go to that 1, just an example. That's definitely an orange. That's a weak positive. And then, okay, and like I said, after that, you would then look at citrate. Is the citrate blue, which would be positive, or is it green, which is a negative? And it's turning blue. Even though the whole thing's not blue, it doesn't matter. It's eventually would all turn blue, but there's the blue slant. So, dramatic conclusion, you started up here, you had rods, you have what's called Bacillus cereus. If the citrate had been negative, you see the catalase is positive, it was a weak orange color here, but the citrate was negative, you would have had Bacillus subtilis. Alright, so that's what we're going to be doing this week, and please see the example on how to write your paper, and we will go from there. So thanks.